Hi, Mr. Lindenberg. This is Liam Poole.
This is Karina Pascalova.
And this is Sophia Davy.
And this is a meiosis video. So to begin meiosis, we begin in meiosis 1. And before we can do that and get into the mitosis process, we have to go into interphase, which is where our DNA is replicating and sister chromatids are forming between the individual chromosomes, which can be shown right here. After this, we enter into prophase, which is where our DNA our homologous pairs start to form, and they find each other and connect in a process called synapsis. Through this process of prophase, the nuclear envelope has dissolved. And then the homologous pairs find each other and link up through synapsis to form bivalents. At the points where they connect, these are called chiasmata, and that is where the DNA recombination occurs and the DNA is crossed over.
The next phase is metaphase 1, and in this case, the centromeres. The next phase is metaphase 1, where the bivalents are going to start lining up in the center of the cell. Their random orientation leads to the independent assortment, which will further increase the genetic diversity. However, the spindle fibers from the actual centrioles are going to start to connect to the centromeres.
During anaphase 1, homologous chromosomes separate and move to opposite plates, while sister chromatids remain together.
So now we're in telophase 1 and cytokinesis, and this is where our chromosomes have ended up at the poles of the cell. And in telophase and then in cytokinesis, a nuclear envelope starts to form to just split the cell in half, and then in cytokinesis, the cell has fully divided.
Continuing on with this process of cytokinesis, this one diploid cell will form these two haploid daughter cells. After cytokinesis, we have interkinesis, which is a short resting phase between meiosis 1 and meiosis 2. No DNA replication occurs during this stage.
In prophase 2, chromosomes condense again and the nuclear membrane breaks down.
One more, Kylie, or two more. In metaphase 2, chromosomes line up individually at the equator of each cell. There, the spindle fibers start to connect to the centromeres of each chromosome.
In anaphase 2, the sister chromatids in the middle will separate to the opposite poles. We can now call these sister chromatids their own chromosomes.
In telophase 2, the chromosomes decondense and the nuclear membrane reinforces.
Now we have cytokinesis, where the cell is going to divide and it's going to form 4 daughter haploid cells.
So non-disjunction is the failure of chromosomes or chromatids to separate fully during meiosis. So in meiosis 1, when they separate, only these, so a full homologous pair came to this side instead of them separating like this. And then in meiosis 2, and these line up and they are separated by anaphase, an entire sister chromatid pair goes, while this separates over here and this goes to the other side. So when it replicates, there's an uneven amount in the haploid daughter cells.